talked about whether, because you know how, like the taste for salt, right? It can, like all your taste buds can receive that, it's that taste, right? But like there's like a bias towards.
Yeah, I mean,
like,
there's a specialization of receptors to a certain extent.
Each receptor, like in whatever area of your tongue, is specialized towards that certain taste. And is it saying that certain regions of the tongue are just more biased to certain tastes?
It's sort of similar to what we talked about for vision, in that you can have cones, let's say for example, respond to different frequencies of light, but they're tuned to a certain frequency that they really like.
Right. Like an on and off kind of do, like the on center.
Well, on and off is going to be a little bit different. It's more about frequency tuning, at least in the context that we had. Let me see if I can show you a slide that might be better suited for that. So said they have different sensitivities. So here is sort of a graphical representation, so you said salty, of sort of where some of the receptors that might be sensitive or sensitized to being particularly good at detecting that chemical stimuli. So all senses are sensitive to the entire tongue, but they're sensitive to different sensitivities. So that's why you see it still might be detected back here, and this is really where it's going to be optimized for detection. And this is sort of a more nuanced, advanced version of the way that people have been typically presenting this material for maybe generation past. If you look at old textbooks, you'll see that a lot of times they just say that there's these spots, let's say, where there's receptors localized for different taste types. And they say that's it. And they don't say that there's a gradient, they don't say that they can be sensitive over the entire tongue. They're just going to say more discreetly, but less accurately. Does that make sense?
Yes.
And so you can have receptors. Let me see if I can show a different picture here. So you can have receptors for different. So these are different cells within a taste bud that are going to respond to different types of chemical concentrations. So sodium chloride, quinine is tonic for you gin and tonic fans or bartenders out there. It's the Q button on the bartending gun. Any of you guys remember weighted tables or anything like that? That's what quinine is. Hydrochloric, sucrose. So anyways, we can see that when you apply those different chemicals to these different cells, that one cell here, cell 3, might really be sensitized to sucrose, where cell 1 might not respond to that at all. Okay? But at the same time, cell 1 and 2 do respond to at least one, if not actually two, stimuli. It's just they respond differently to those stimuli. Does that make sense? So it can so you're going to have sort of specialization where one type of receptor is going to prefer a certain stimuli, but it's going to be, in the context of it's going to prefer that for maximal activation. It might still have sort of lower activation, that for a different stimuli. Right. In the same way that we talked about vision, working through this for a second. If we looked at, we draw this out, let's see. So when we had our different frequencies of light, right? Remember we had tuning curves for colors, right? Might look something like this. The orientation of these, in terms of which one comes first, red, green, or blue, might be different. I'm just casually throwing them up there for illustrative purposes. So, what does this mean? What was? Do you guys remember what was talked about in terms of that reflecting?
I don't know the image, but I just.
So so we have, right, the easiest way to start, right, when we can't figure it out, is to figure out what's being shown. So if we're talking about graphs, then the first place to start would be thinking about axes, right? Okay, so what's on our x-axis? Frequency, exactly. So frequency, to get even more drilled down, frequency of what?
Wavelength.
Wavelength. Color is the frequency, is the wavelength frequency. So you're right, but we can, it would have been circular if we said that. So so we basically wavelength, right? That's what usually nanometers or something like that. Okay, so then what do we have here? What are these tuning curves of?
Would be the responsiveness to a certain frequency, so the average.
Responsiveness of what?
Yeah.
Color.
To light, but of what?
Color.
Yeah, yeah, so it's a cell's response properties, right? So it's the cell's responsiveness. So, what should we call that on the y-axis then? So let's just call this responsiveness. Of course, these are cones, because our rods would not be the color side of the frequency. So we have different tuning curves of different cone cells, right? So you have so A, B, and C. Yeah? Okay. So what does this tell us about these cells?
They're more responsive to different certain wavelengths of light. And certain cones are responsive to certain wavelengths of light.
Yeah,
They have a range of responsiveness, and as that range overlaps.
exactly. So we learned one thing, we learned two things, right? So you said, first thing we learned is that different cells have different preferences or different sensitivities, right? And so at a general level, that's our first step, right? It just says different things like different things. Of course, other than that. And what was it that you said?
I said they have a specific frequency that they respond to the best, but they also have a range of frequencies.
Exactly. So it's not just that we can restate what he said, right? Which is that different cones have a frequency that they respond to the best, which is right. This is their best response. So what does this mean? It just means that it's this point, and this point, and this point, because this is the point there, this is that point, and that point. This is the wavelength for each of those that it responds to the best. But you made a really, really, really good insight, right? Which is that we did not draw bar graphs here, did we? Why did we not draw bar graphs? Yeah, okay, so overlaps the third thing, we'll get to that in a second.
They're using a continual wavelength is a continuous variable, so there's spread to what it will respond to.
Exactly. So if all this cell cared about was a wavelength of light at this x, I don't know, y, or z, exact now here, then we would just have a bar graph. It would just be one thing, whoop. But that's not what we drew because that's not what's real. What's real is that not just do they have a specific frequency that they really, really like, but they're still going to respond to other frequencies beyond it, just at a lesser degree. So at this point in the graph, it would be responding to that one. At this point in the graph, right? So we don't draw all those bar graphs next to each other, we just draw the top of it. So we learned that there's specificity, but there's also sort of genericness as well, right? So it's two different pieces of information. And the third one that we can learn from this is that there's overlapping elements, right? Which is that there's some frequencies. Actually, this is a really good example here. At this frequency point, you're going to get cell A, cell B, and cell C all firing, won't you? It's kind of crazy. I thought that we had specific types of, and this is all
Yeah, it's all related to taste.
This is all the same.
Instead of wavelengths, you could use different tastes.
Yeah.
I see what you're getting.
So that's why we went down this road, is because, like I said, it's illustrative of this same point that we saw in terms of population coding or coding of different cells that are getting information, because you can now say A, B, and C and look at their recording traces, right? So let's say cell A at this wavelength nanometer equals Y, because that's just the value we gave it there. Okay, just keep it simple. We don't need any numbers right now. And A is going to be activated, which is a little bit of a baby bit, right? Okay, so A might look like maybe like that. You think? A couple action potentials?
Yeah.
Well, action potentials?
Mhm.
Cone cells?
No, it would,
They don't do action potentials, do they? Yeah. So,
Tricky.
it's tricky, isn't it? Alright, so you might see little bloops. And actually, since they're cones, let's be accurate. Let's make them this way, right? Okay. Right, because they're going to hyperpolarize, not depolarize.
Mhm.
So, B, what would this one look like? It's going to be a little bit more, right?
Yeah.
So, right, and of course we've got C, or actually B would be like a green cell, right?
yeah.
Okay, so
Yeah,
these are
B
gonna
is
be,
kind of
let's make these really big. And then we'll make the C a little bit somewhere in between. Right, because that's the red line. And so, when you have these neurons projecting onto, let's say, another neuron, right, that's receiving this input, which is, of course, anatomically going to be bipolar cells. We're just going to have these signals go through the bipolar cells, and let's say have it be your retinal ganglion cells, right? This is going to detect this different pattern. And so it knows what the color is, because it's not just that this one's firing maximally, but it's the combination of this and this that allows it to detect the difference between firing really close to maximal here versus this, because they're all going to have a slightly different balance of that community of three different cells sending relatively stronger and weaker signals. Make sense? So the same idea is going to be evident in chemical taste. Is that combination? We're talking about convergence. So that's kind of what I'm seeing there. Really good question. Okay.
That was awesome.
Oh, thanks.
I like that. Oh, stop. You're like,
Come on.
you're like the cat in the TV. trying to, yeah.
They would just get another one.
Oh, boy.
Anybody think that was real?
No.
No, we're just watching you be weird.
Alright. Okay, who can draw me something from the olfactory system?
Olfactory.
Like colors instead?
You can use that. Like 11 rings. So I could do like the papillae or whatever it's called, where it's got like the.
That would be the taste system, not the olfactory system.
Oh.
He's like,
Well, dang.
ah.
Can anybody help? Yeah?
I'm sure do you want another one of those graphs or just something?
Something.
I'll draw something.
The board is all yours. And let's help her if there's spots that she can be helped.
Corey, do we hold your binders? Do you not have to keep like moving back and forth?
Maybe.
Can you talk us through what you're walking us through?
Sure. So, okay, so this is actually the, here, hold on, I have to draw some things first, so that. Okay, this is my ugly cerebellum right here.
It's beautiful.
Yes.
Don't let anyone tell you.
Yeah, it's in the eye of the beholder.
And then here is the, make it green, the base of
Well, that's okay.
it anyway, not to scale, no.
It's a cute.
That's just the side of it, it's not that, the whole.
So what angle view then would we be looking at? now?
This would be
Sagittal.
Thank you.
Tickle my throat.
Thank you. Ta-da.
Okay, so where is the, anybody know where the olfax is anatomically? Like where's some areas we might call that? Alright, we're gonna talk about that a little bit today. We have an olfactory bulb and an olfactory tract. Okay, and where are they leading to? Yeah, eventually it leads to the olfactory cortex. So this is,
It's gonna go through the thalamus.
the thalamus is gonna be a player in the conversation, so we might have, alright, so if this is our, let's see if I can add here. Alright, there's our lips, there's a chin, we'll give him a mustache,
Okay.
okay?
I thought you were gonna give him a nose here for a second.
We can do, no, we're gonna look at that too, so.
Oh, no,
Alright, so here's the nose and our olfactory bulb, right? We're gonna spend a lot of time talking about what's happening here en route to the olfactory bulb. And we'll talk about the pathways of where we're gonna move from the olfactory bulb to the tract, cortical areas, and some of those same signal processing elements that we've talked about now, vision, audition, vestibular systems. So now we're moving into a different sense, smell. Also incredibly important. I haven't decided yet which sense I think is most important. Because we talk about this every semester, I've been doing this for a long time. I always try to underscore how important each sense is, especially ones like we talked about last class, vestibular or olfactory, really think of, but presumably take for granted a lot, you know? And, I mean, I wouldn't want to lose any of them.
So if you had to keep one, what would it be?
I don't know.
Sight.
I want to be able to see where I'm going.
But pizza.
Yeah, that's it. Would you, I mean,
I'd rather have, I would rather have colors in my life than taste.
Oh, that
I'd
is
rather
poetic.
yeah, what about like good wine and food and cake?
To get more
And-
from food with smell than you do with taste.
Yeah.
I also
I mean,
like to
you
hear
really want-
someone sneaking up on me
Yeah,
in the middle of the night, like.
Oh, wouldn't you like to be able to smell someone sneaking up on
No.
What?
you? No? So you said smell might be your most important, right?
Yeah.
Why?
I mean, for me personally, because I feel like you can smell different things. You can smell people, you can smell weather, you can smell like if there's a fire, you can smell you can like sense a bunch of different things that you can kind of see with your eye, with your nose. And I feel like the sense of smell is really sensitive too, right?
Yeah, it's really
And
sensitive.
it draws a lot to memory too, a lot more to memory than
Oh, yeah.
the other one, so.
Big time.
it-
Big memory.
So if you smelled something that was at first and you couldn't see it, and then you smelled it again, you would immediately know.
I can still remember the perfume of like my first girlfriend in high school. It was really good. There's a strong memory link, right? For obvious reasons of evolutionary importance and stuff like that. But it is a really important sense. Let's think about some of those things. Let's think about times when your senses might be dull, right? So, ladies and guys, imagine maybe that you're out having a good time at some really crazy club, and it's just like thumping. Right? It's like, right? So I went to college in Jersey. We know how to pump our fists. And so let's put yourself in that situation, and your senses become dull because you can't see much because it's dark, right? You can't really hear much because you've got some crazy DJ on who knows what kind of uppers just blowing up the speakers, right? And you hear there's all these sounds and music and people talking. Can't really hear much, right? People try to talk and you're like, oh yeah, sure, okay, right? You pretend like you hear what they say, but you can't. And then when you have to talk to your best friends, you have to like yell in there. You hear, I want to go to the bathroom. Okay, well, we'll go. And right? So your senses, right? We already know that they're saturated, so they're not going to work that well to be discriminative of information in your environment, right? And so, let's say you're out there on the dance floor having a good time, and you just get your swerve on, and there is, so your boyfriend starts to come up to you and starts dancing with you. And so you guys are having a good time, doing your thing on the dance floor, and you recognize him because of the memory of that cologne that he wears, right? But what if you find out a little bit later on that there's more than one person who bought that cologne at the department store or at Walgreens that night, and you're laughing like you this isn't the first time you've heard this story.
Friday night.
Right, but anyways, the point is right, like, what if your smell lets you down? And you thought it was your boyfriend the whole time behind you, and it's some other total creep show having a really good time. Right? You need the sense to work for you. And you need to have it work beyond just a single scent of whatever was on sale at CVS. And you know what I'm saying? We need to be able to discriminate those different smells that are going to come at the same time, perhaps. And let us know when to turn around and smack someone for shouldn't have been dancing with us. So you know, I think smell gets undersold as a bad rap. I think it's a really one of the key ones. I don't know if you want to lose, because then what if you're doing something you shouldn't be doing? The signal to that is that it's really funky smelling.
Oh, no.
You should get out of there, right? You shouldn't get so close to whatever that thing is. And like that's a very evolutionarily important signal, right? Now, what if you lost that one because you wanted to keep your sense of vision or something? I mean, I don't know.
Now, being a taste and what to do with smell, if you lost, could you entirely lose your sense of taste if you still have your smell, or would it still be there a little bit?
I think you'd probably still have it in a little bit, but you'd lose some of it. So
Maybe to take the sensitivity part.
I think so. There's a couple examples if I can remember them off the top of my head, and they might need some clarity with some follow-up digging. But I believe that you cannot tell the difference between a potato and an apple if you bite into each of them with your nose closed.
Same with onion, I heard.
Yeah, or maybe an onion.
Maybe it's
should
an
have stayed.
onion. They're like, yeah, they taste exactly the same. And I'm like, I don't know what you're talking about. I don't think so.
So anyways, the point is that the senses are, as you mentioned, chemosensation, which is taste, and olfaction, which is smell. They're very closely coupled, but they're also independent senses too. And there's times when they're going to overlap and whatnot, but they are important. And I just wanted to underscore those elements as we dive into it, because sometimes we take it for granted. We can't imagine being blind or deaf. That would be, those are easy things to think about, losing that sensation of. But, I mean,
What about even touch though? Because I think it's also key.
yeah, so some touch we'll look at in a couple lectures from now. What we call somatosensory information. So sensory information from our somato or skin, body, and it's one of the things that if you press me on it, like I said, I haven't figured out which one I think is most important. Right? It's like trying to figure out a child, but I might be pressed to maybe say somatosensory system could be my favorite, or that I think maybe most arguably important. It's completely just a subjective opinion issue, right? Because they're all really important. But when we talk about the somatosensory system, we'll talk about the importance of that with conversations like this too. And think about if you'd want to live your life without the sense of being able to feel some really good things that make some pretty happy instances in life. Okay. Right? I mean, there's evolutional, I mean, arguments to make about behaviors that enter the conversation. Somatosensory is a very, very good, important system too. So anyways, olfaction. We're going to talk about a lot what's happening in this sort of cellular and molecular layer here, which is where we'll see that the information of olfaction, which is smell, it's not chemosensation, which we refer to taste, but it's going to be a gaseous chemical. So obviously it's something we smell in the air. So it's going to come up through the nose, no surprises there, but it's not the nose that smells. The nose is just a cavern that collects it. It's kind of like, it's not what hears, is it? You guys are all experts at that now, right? So if I ask you, how do we hear things? Your answer should not be the ear, right? Because it's just a channel, right? We have opinions just grabbing that information and just channeling it into the other mechanisms that we spend a lot of time exploring. So same thing here. The nose is just it's just the tissue that channels that information. And of course, you're going to have a lot of hair and mucus and all sorts of goopy, gross stuff in here. That's going to be very important for a couple different things. One, it's going to help grab those chemicals in the air, but also they're going to filter out the toxins. It's really, really important things. We've talked about senses and things like that. Has we lost vision here?
You said you lost your
Yeah, I've lost times.
Can we hear that story?
Yeah, I had to get ear case surgery on my eyes. So So PRK is it's a old surgery they used to do in the Navy, laser vision. So before they have LASIK, you have laser vision. And so when you have the eyeball, let's say, you have your eyeball, right? We're looking at it straight in. You need to reshape the cornea, because remember we talked about light hitting that? And when it's, you get nearsighted or farsighted essentially, because it's displaced on the lens and where the cornea is shaped. So you use a laser to go reshape the cornea so it's better adapted to that refraction of light. And so that's what you do. And so in LASIK, you basically cut essentially a flap in the eye, and then you pull it backwards. You pull the flap over, and that allows the laser to then go in and do its work to reshape the cornea. Now, that's a really nice advance because it's essentially relatively painless. And then once this flap is pulled back and you use the laser to go in there, the flap is basically your epithelial. It's a clear layer of skin essentially. Then you can put the flap back over it, and there's not a lot of blood vessels there, so it doesn't actually ever heal because it's the physics, surface tension. It holds that flap in place really fine. That's why LASIK's a nice thing. PRK, on the other hand, is what preceded that. So in the 80s and the 90s, the Navy did a lot of experimentally. And instead of having a flap, what you would do is you would just basically use sort of like a razor blade, as you might think of it, and you just scrape off the top layer of skin, and it grows back in about 3 days. But it's really painful when it's back because you're blinking every time, and your eyes actually exposed without break that layer off, and now the laser can go in. So the surgery, I just had to get that done because I was really had a bad place. I was a super dork. I had really fast. So it was a big dork. So yeah, and while I would play football, and I was in the grunt, there's no grunt. Yeah, I had really long. I played football in my seat either. I learned how to wrestle without seeing because I had to take my glasses off when I was blind. It's just like blurry stuff. And the hair was in the way, so anyways, that happened. And once I finally got health insurance as a postdoc, I got that surgery, but it puts you to sleep. And then for about a month, you see stuff clearly. So, yeah, couldn't see. And it turned out it actually really helped me as a kid, though, to see that. When I was a bouncer, I got used to people attacking me without see and manage it. And then I ended up working closely with a team in college who was fully blind. Friend with this guy, so realized like he actually heard Russell, like the blind guy in high school and in college. He's a big, jacked up dude. He can't see peas. He can't do anything. So I mean, he has like somebody's always helping him. Like he's full out handicapped. And so I what?
While you pee.
Well, you're gonna go this way. You're gonna go this way. I mean, you know, if so, I ended up kind of being his caretaker during those years of school, and we would train together, and it helped me to be able to coach and work with him because I could understand how he was doing it. And then when I had high school kids, I took the high school team, and we'll get back to this in a second, but they weren't listening to me one day about how I was teaching how you have to plan ahead, whether it's school, but in this case, kind of like combat situations of wrestling. Can't just do your move. You've got to know what their defense is going to be. It's like a chess game, and you got to know what your follow-up response to their response is going to be. You've got to plan these steps out. You've got to feel where your hip positions are so that you can accommodate that situation. You're always thinking ahead, planning ahead, and you have to know what's going to happen next. And you have to, the whole nature of the game, just like chess, is anticipation. If you're not doing that right, you're going to lose, and vision has no part of it. So they weren't listening to me. I said, all right, fine. Took my shirt off, wrapped it around my face, blindfolded myself, and I said, all 4 of you guys over there, just come attack me. They're like, yeah. I'm like, come on, guys. So I'm like, all right. So they came, and I was able to work them pretty easily because the illustrative example was to underscore how you don't need vision. They didn't need to see because it's about somatic sensory information, anticipation, planning ahead, and you can handle this. So that was that and the and then that surgery gave me they call them? Some other type of vision problem that they're gonna have to do again. Decide again the next year and get it done again, which is no fun. So blind twice, plus other kid. Yeah. But bad luck to the people who attack in the dark. So, anyways, so we were talking about, how do we get to that? What were we finishing?
You were
We were talking about the alphabet.
Oh yeah, oh yeah,
The
because you
importance
can use
of-
yourself. So I said anyone lost vision. Some of us might have lost hearing before, perhaps if you ruptured an eardrum or something happened, or you know, had times when we can't hear anything, right? We can actually see something called anosmia. So it's called people who become anosmic, and it's not totally uncommon. Smokers, for example, lose a lot of their sense of smell. Not entirely always, of course, but they can lose, just like we talked about those frequency bands, those tuning curves, or the taste receptors in certain frequencies. You can lose your ability to smell certain odors. But fortunately, it can regenerate and come back. So the regeneration abilities of these neurons that we'll take a look at, particularly in this olfactory tract involved in the system, are one of the few places in the brain where their neurons constantly regenerate. So when people say, once you lose brain cells, they never come back, not true. At least not in definitionally true. It can be true in some areas of the brain, but these brain cells, these olfactory neurons, they regenerate, what you call neurogenesis. And that's why we can gain those senses of smell back, maybe after a few weeks. If you had somehow you got exposed to really bad ammonia or some toxin, right? Basically destroys your sense to smell certain things. But let's say that you've made the smart choice to stop smoking. Maybe about a month or two later, you start to smell some new things that might have otherwise been blocked out. Because these brains, or the neurons, are starting to regrow and regenerate. And we'll see that they have different receptors, different genes in them that express different proteins for receptors for different odors that now can actually be utilized. I'm sorry, there's a hand up.
Let me see, is neurogenesis fairly a novel theory or
Yeah, relatively speaking. You go back to the days of like the call crowds and 100 years ago, 50 years ago, there's textbook dogma. Once you lose brain cells, they're gone, right? And for good reason, they thought that. I mean, it's tough to see new brain cells being born in real life, right? I mean, we think of the tools and technology needed to show that. It's very difficult. But it's probably about 20 years ago or so. Rusty Gage did a lot of the pioneering work at UC San Diego, and some other people have as well. But yeah, it's a relatively new phenomenon. When I started teaching this maybe 10 years ago or so, it was a lot easier to say that there's these new discoveries that just came out and they're showing we can regrow brain cells, but I can't really say that anymore because it's becoming longer in time and it's definitely more established and recognized. But it's a really phenomenal area of interest, and particularly the ability that characterize these olfactory receptor neurons that we'll look at, and which have these neurogenesis abilities, gained Axel and Buck the Nobel Prize in 2004. So again, another sensory system characterized and recognized for that characterization with the prize from Stockholm, which is where they give the Nobel Prize at. It's a city in Sweden. Later.
So you actually lose the ability to smell certain things, not just affecting the neurons, but just to not smell anymore because you become accustomed to it, I guess? Like
Anosmia?
Yeah. So I was just trying to figure out the difference between, so anosmia is where it actually affects the neuron, you lose the ability to.
Anosmia just means can't smell.
Oh, okay.
There can be different reasons for that, but usually it's because some toxins come in and destroy those receptors.
So like people that live like next to a dairy where they don't smell that smell anymore, would that be a form of anosmia, or would that just be used to it?
I would consider it a form of anosmia.
Okay.
But that's just me. Let's see here. Okay, so anyways, what we're talking about here, I'll show you. I'll walk through some of the figures from the from your text, and then I can show you some other figures as well if you want. Maybe help to clarify it, but let's see here. So, oh, thank you. Okie doke. So by the way, I don't plan on testing you guys on chemoreception taste, by the way. So that's not in my plans, unless you want it to be. If you like that content and you thought it was cool and interesting, we can do that, that's fine too. But it wasn't in my plan. So that's the first half of the chapter, but we'll jump to kind of the olfactory side. And so the ideas here, we've already talked about some of the benefits in terms of warning of harmful substances or dudes at the bar. And of course, we talked about it combining with taste for identifying foods, poisons, all sorts of other things. There's other elements of smell that are less well characterized in humans, but are well known in animals, known as pheromones. So they're basically things that can serve as a form of communication, particularly useful for reproduction and aggression, submission, things like that. In the animals, it's processed through something called a vomeronasal organ, but in humans, we haven't really found an organ like that works. There's some behavioral evidence that people can be responsive to things like pheromones, but we don't really have a mechanism real well characterized that's been able to explain that yet. So for the longest time, there was sort of a big debate about whether humans even are they completely insensitive to pheromones. This is just nonsense. And for the most part, the evidence has been really tough to show that pheromones affect humans. There's some stuff that's come out that's kind of made a case, but a lot of the big claims about it are essentially made by perfume companies who have what you might call a conflict of interest. But it doesn't mean that there might not be something there, I guess is the point. And for the most part, we'll kind of leave the vomeronasal organ alone from on the human side. And we might see some elements in animals. So this is more of a closer picture of I think what Corey was trying to draw. Is that right, Corey? Yeah. Okay. So it's pretty close. And so here's our nose, right? So this is a sagittal view, essentially, me looking this way, right? And so we have air coming in through the nostril, okay? And as it's coming in, it's singeing the nostril, right? If you're on Burgundy, and it's a bad smell, it singes the nostril. It's quite pungent, right? And so it's coming in there, and just for context, so you know what we're looking at, right? This is our nasal cavity. Okay, this is the lip. Okay, this is the tooth. Okay, this would be the mouth, probably in here somewhere, right? Food's coming in, it's getting chomped on. Pearly whites. Air's coming in here. This is where all of our action's going to be happening here. If you want, you want to hit the light? Is it easier to see if we turn that off? Is that better? Yeah. Okay. So we're going to look into this particular area because this is where you're going to have a lot of mucus and membrane, and it's going to be very helpful in terms of protecting these very delicate and very important receptors. Okay, particularly from the toxins in the air that would otherwise kill them and destroy them. And if you have those destroyed, you're not going to be able to tell who it is sometimes that might be dancing behind you in the club, right? And sometimes you can tell quite clearly if it's somebody who smells quite different from the person who you were supposed to be dancing with, right? And so we need those suckers to work really well there, okay? And the toxins would otherwise destroy them. We draw upon those mucous membranes and other types of protection to help stave off against that toxicity. So this bone here, this is the brain, by the way, orbital frontal cortex. It's the ventral part of the frontal lobe. Okay, and it's separated from the nasal cavity by this bone in the skull, right? So this is your skull, and the bone is going to come across here. That bone is called the cribriform plate. Okay, now the cribriform plate, if we look at it, really zoomed in, right, grab this little snapshot, zooming in over here, this is our cribriform plate. It's bone. But what's really cool about it is it's got spots that the neurons grow through. So this is one of those sort of special situations, kind of like vision when we talked about neurons in the visual system are sort of special because you got these neurons in the retina that are actually outside the skull, right? So there's access, right? And it was one of the reasons why it was an early studied system historically, and why so much of the field of neuroscience historically is based on research from the visual system. So it's the most easily accessible. You get at the neurons without getting through the skull. Well, here, the neurons are getting through the skull, right? Now, they're connected to the olfactory bulb that Corey nicely drew us earlier. And the olfactory bulb is going to project through the olfactory tract and enter into the brain and carry that information that's coming in and singeing the nostrils. Okay? And so we're going to take a look at the processes by which that information is transduced. Okie doke. Sound good? All right. So, when we take a snapshot look at this, we can see that not only does the neurons go through that cribriform plate here and into the olfactory bulb, okay, but we can take a look at the shape of the neurons. It's maybe a good place to start, okay? So we've got our nucleus here, our soma with the nucleus, okay? We've got its axon projecting into the brain, and then we've got its dendrites out here, right? The proverbial feelers, sensing stuff in the environment. And they look like little hair cells themselves, right? We call them cilia. The cilia of our olfactory cells. Now, our olfactory receptor cells, or olfactory receptor neurons, you're going to see potentially abbreviated a lot, it's ORNs, olfactory receptor neurons. So in case you see that, that's what it's referring to. And, right, they're not just, no cell is an island, right? It takes a village to raise a child. You can't just have a cell here, you need a bunch of supporting cells to hold it in place. So you're going to have these supporting cells. Sometimes you'll see them called sustentacular cells. But they're going to be there, and then you're also going to have these basal cells, and you're going to have these Bowman glands as well. And Bowman glands are basically going to be cells that are going to be producing the mucus and stuff like that. So excreting that stuff that is going to be helpful. And so we're going to look at first what's going to happen here, how that signal sends up into the brain. Sound good? Any questions so far? Okay.
Go ahead.
Jay.
Oh, I was just gonna say, what did you say, the OLN?
ORN,
RN, okay.
olfactory receptor neurons.
And then what were the names of the cells that produce mucus?
The what?
The cells that help production of mucus?
Bowman glands.
Bowman?
Bowman, yeah. B-O-W-M-A-N. I believe we'll have some pictures of them later. So, we talked about anosmia being that inability to smell. We talked about the cribriform plate already, that bone through which the axons are penetrating. And we'll look at this transduction process just like we looked at the transduction process in vision and audition and vestibular system detection. And it's important to kind of note, right, that we're relatively weak on the scale. So we started off the conversation today talking about the importance of smell and evolutionary elements of it. And, you know, we're not very old on the evolutionary tree, right? We're pretty young. So it's a very old process evolutionarily because it's so important. And really, if you look at some of the older species, rodents, sheep, all sorts of animals, dogs, they have way, way, way more advanced systems than we have because it's such a more important sense for them, you know, to be able to smell stuff. And so they have billions more neurons for odor detection, and they're really pretty good at it. So, and you can see different cortical representations of olfactory cortex in the different species, depending on how important that sense is for an animal's survival. Okay, so let's see here. When we talk about this transduction process, we're going to look at these dendrites, kind of at the side of the cell, and particularly the cilia. And now they're embedded in this mucus, sort of just hanging out. And what's going to happen is we want to see that there's an action potential that gets shot up here through the cribriform plate into the olfactory bulb. Okay, it's going to have to be generated in our soma, which means we're going to have to affect some type of depolarizations down here, right? So all these as a framework is all very similar to what we talked about with action potentials, vision, audition, all these types of things. We need to change the membrane potential to elicit action potentials. And we're going to look at some of the ions, the ion channels, and some of the systems that are involved in that, okay? So the basic idea is that you're going to get these chemicals in the air, odorant molecules, okay? They're going to bind. They're going to bind to a G protein coupled receptor. Okay, in this case, G-olf. G protein for olfactory system. So you'll see it abbreviated as G-olf. G-O-L-F, basically. And it's a little subscript there. Okay, so you're going to bind to this protein. It's not an ionotropic, it's going to be a metabotropic type of situation where you have this G protein coupled reaction where it's going to activate adenyl cyclase, which when combined with ATP, which is our energy source in the cell, right, it's going to lead something called cyclic AMP, which we talked about earlier. Cyclic AMP is going to then come and bind to some ion channels that's going to allow sodium and calcium to come in. And interestingly enough, on a unique side of this system, we're going to have chloride efflux, which means that there must be an abnormal concentration gradient of chloride if it's being driven out of the cell, right? But it effluxes, which means it really gets very positive, right? Because you're losing a bunch more negative charge.
So for this, calcium and sodium would both come in through the same channel?
It certainly looks that way in this diagram, although I cannot personally vouch for that.
Because it looks, according to this, it looks like this calcium comes in anyway.
Yeah, it certainly looks like that, doesn't it? It's not going to be, knowing that one way or another isn't going to be a component of what you'll be tested on, I can promise you that. To be honest, I wouldn't be surprised if it were either way. If somebody told me that they have their own channels, I'd say, yeah, that makes a lot of sense. If somebody told me that the size of those ions are sufficient enough to fit through the size of those channels, I could probably not be too skeptical about that either. I don't know the detail of that. But the point is that we have depolarization and we have a little extra oomph to it, right, by the efflux of the chloride, and that makes this really depolarized. And so when that happens, we're going to get our graded potential, just like we talked about, right? Our graded potential is going to turn into eliciting action potentials in the soma. You can see those action potentials carried out through the axon there. Make sense? That's the basic idea. Thanks. Saved it. Okay, so here we're looking at a mouse version of that, and what we can see is that there's different genes for different receptors. Okay, so we're looking at that sort of side view here. We can see different colorations showing that there's different distributions of genes, gene categories, and these gene groups are going to be involved in producing different types of receptors. So there's thousands of different types of receptors that are molecularly designed to be responsive to only certain molecules and not other molecules. So this is an extremely sensitive version of what we started off class talking about. The chemoreceptive cells, the cone cells in vision, right? Optimize sensitivity to certain stimuli and not others. Here we have thousands of different olfactory receptor neurons. Remember, this is a nice, you guys might not agree with me at this point, but I think this is a very nice cartoon, right? But that's all it is. This is a very nice, simple cartoon. You guys might think it's simple, but it is, right? Because we can always go down the rabbit hole and look at how crazy complex it is, but we hold off on that. And this is just a little green blob.